Hello, my name is Mauricio Cáceres. I'm a PhD candidate at the lab of Dr. Sierra at the University of Puerto Rico School of Medicine. Today I will be talking to you about my work regarding the effect of glyphosate on anxiety-like behaviors and fear in rats. Glyphosate is the most common active ingredient in commercial herbicides, mainly Roundup. Though glyphosate was often thought to be safe for animals and humans because it attacks a metabolic pathway almost exclusive to plants, recent epidemiological studies have found a correlation between the increased use of glyphosate worldwide and the increased incidence of neurological and emotional disorders such as anxiety. This has even been supported by studies exposing animals to glyphosate, showing increased anxiety-like behaviors. Interestingly, despite the fact that the brain circuitry that promotes anxiety is very similar to the one involved in the expression of fear, the effect of glyphosate on fear has not been studied. Therefore, the aim of this study is to evaluate the effect of systemic glyphosate on fear in rats. We hypothesize that given the similarity between the brain circuitries, intraperitoneal injections of glyphosate will lead to increased fear expression in rats. To test this, we exposed adult male rats to 100 mg per kilogram of glyphosate via intraperitoneal injections every 2 days for 3 weeks. Rats were then tested for anxiety-like behavior using two paradigms: the open field test after 2 weeks of exposure, which consists in placing the animal in an open box and allowing the animal to explore the open box, where the areas next to the walls are considered to be low anxiety areas, and the area within the center of the box is considered to be high anxiety areas. And also, we've measured the anxiety levels during a light dark box test. In this paradigm, what we do is place an animal inside of a box that has two compartments, a dark compartment and a well-lit compartment. At the beginning of the experiment, we place the animal in the well-lit compartment and allow it to explore. Rats have a natural tendency to prefer areas that are dark and seem more safe to them, where they are enclosed and have a lot of cover. And they do not like areas that are well lit. Therefore, the area where the light bulb is considered high anxiety, and the area where the darkness is considered low anxiety. And finally, after the exposure, rats underwent auditory fear conditioning, which consisted in exposing the animals to a tone that was paired with a shock. The next day, the animals were tested for fear memory by presenting them to the same tone, but in absence of a shock. What we did to measure fear expression here is we measured how long during the tone did these animals spend freezing. Freezing is actually a normal behavior in rats and is used to express fear. It consists of absolutely no movements except those that are required for breathing. Now, when we performed the open field test, we noticed that, as you can see in this example of a tracking of two animals, a control animal and a glyphosate animal, glyphosate did in fact lead these animals to avoid the center of the open field and spend most of their time traveling or exploring the borders and corners, which are considered low anxiety when you compare them to the control animals. When we quantified the data, we noticed that glyphosate actually significantly reduced the amount of time that the animals entered the center, the number of times they entered, the amount of overall time that they spent in the center, and also the distance traveled within that center. Together, these indicate that glyphosate does increase anxiety-like behaviors in the open field. Next, we analyzed the dark light box, where we noticed that despite there not being a significant difference in the number of entries to the light, glyphosate showed a strong tendency to reduce the number of entries. This held true when we measured the amount of time spent within the light, where there was no significant difference, but again it showed a strong tendency to reduce the amount of time spent in the light. Interestingly, when we looked at what's called the latency to first exit, which is when you place the animal inside of the light area for the first time and you just count how long it takes for him to actually escape or to run away from this well-lit area, glyphosate did significantly reduce the amount of time it took for them to decide to run away. So that may indicate that, at least in this specific parameter, glyphosate does seem to increase the expression of anxiety-like behaviors. When we performed and analyzed the effect of glyphosate on fear, we noticed that it did not affect either fear conditioning or fear memory. However, we must point out that a problem with our experimental design may exist, where repeat injections could be considered a prolonged stressor. and prolonged stressors have been shown to increase fear expression, and this may have confounded our results. As you can see here in Figure A, both of our groups, the glyphosate groups and the control groups, are showing fear expression levels, or percent time freezing, above 85%. Moreover, when we collapse all of the time spent freezing during the fear memory test, the control groups are well above 85%, and the glyphosate groups are just a bit higher than that. Now, the fact that our control groups are this high might make it very difficult to actually discern or not whether there's a difference or whether this glyphosate actually increased fear expression. So that would make us need to reanalyze or reassess the method of exposure that we would be using. So in conclusion, it seems that systemic glyphosate does increase anxiety-like behaviors in the open field and light dark box, but may not affect fear expression, at least in the way that we tested these animals. So for future directions, we will be looking to assess alternate routes of exposure, such as maybe oral exposure through drinking water or oral gavage, as well as perform immunohistochemistry for neuronal activity and using CFOS in brain regions that are important for anxiety and fear. These are my references for this poster, and I'd like to thank all of the members of the Sierra Lab and our funding sources for this opportunity. If you have any questions, I look forward to answering them. Thank you.